Hello, my name is Topolsky. I work as a software developer at the Swiss Institute of Bioinformatics, and in this poster presentation, I'm going to show the work that has been done in our computational biology group at ETH Zurich on VPipe, our viral genomics pipeline, as we are currently applying it to SARS-CoV-2. RNA viruses have characteristics which have impact on the dynamics of their evolution. They have higher mutation rates, they have short generation times, and exist in large population sizes. This gives rise to both inter-host and intra-host genetic diversity. Because of the additional resolution it offers, next-generation sequencing has been the method of choice to measure this diversity. VPipe is a pipeline that has been created to assist in the processing of NGS data. VPipe takes as input the raw reads coming from the sequencing machine and a reference from the virus being studied. It can perform quality control, it does align the reads, it generates consensus sequences, and then calls the SNV against these sequences. It can also generate haplotypes, either at the global level or local level inside the windows. It is implemented in SnakeMake. This helps installing automatically the component of the pipeline. All these components are available under open source licenses and can be obtained as packages on Bioconda repository. This includes both our own component, which were developed here at the CBEG of ETH, such as, for example, a specific aligner for RNA viruses and a variant caller which relies on local window to increase the quality of the output, and also the third-party component that we are also using. Such installation has been simplified with a single command handling the installation. A tutorial shows its use and shows the usage of VPipe using SARS-CoV-2 as an example. We have also an interactive visualization report which helps people looking at the virus SNV look how they map to known annotated regions of the genome or other regions of interest, such as, for example, primers, and the SNVs can be filtered by quality levels. One of the current applications of VPipe is using it to automate the sequencing of SARS-CoV-2 genetic material. Swab-tests which have been detected positive by the Viollier laboratory in Switzerland have been obtained by the Genomics Facility in Basel. There, sequencing analysis is performed using an Illumina MiSeq, and then the raw data is uploaded onto an OpenBIS repository. VPipe takes care of automatically downloading the raw reads from OpenBIS, performing the analysis, and also doing quality checks and making reports to assess the quality of the results. The consensus sequences are then made available to the computational evolution group for downstream analysis. The sequences are released through GSAID and also on the European Nucleotide Archive under project PRG-EB38472. The raw reads will be available soon on the Sequence Read Archive under the same project. This open release enables NextStrain to draw a phylogenetic tree of the sequences found in Switzerland. Currently, 3/4 of the Swiss samples visible there have been processed by VPipe. These sequences have now joined the amount of information which is available from the Swiss National COVID Science Task Force to help getting a better picture of the evolution of the disease. The Swiss cohort has also been contributed to the collection of European sequences which have been analyzed and published in Eurosurveillance. Out of the 713 sequences for Switzerland contributed to this publication, 681 have been processed with VPipe. The Computational Evolution Group of ETH is publishing an article using phylogenetic trees to estimate the spread of SARS-CoV-2 virus. It's available as a preprint. Using the intra-host analysis possibilities offered by VPipe, the Computational Biology Group of ETH is also publishing an in-depth analysis of the genetic diversity of SARS-CoV-2 within patients. This publication combines the Swiss COVID-19 cohort and also nearly 4,000 deep sequencings available on the Sequence Read Archive. Preprint is available in bioRxiv. And with that, I would encourage you to come visit our website. You will find a mailing list where you can get subscribed to get information about new releases and other such announcements. You can find email address to which you can ask questions. You'll find tutorials and much more information about VPipe. I want to thank all the developers on our team who have contributed to make VPipe, and also want to thank all the external collaborators with whom we had opportunity to work with and to develop our current technology. Thanks also to all the past members who have helped in developing the previous versions of the pipeline. Thank you for listening to this poster presentation.